Hi, this is Miss Litton, and this is my wonderful 7th period review team here. Say hi.
Hi.
That's weak. Anyway, we are talking about the neuron and the nervous system. You want to just go through and kind of review the evolution of the nervous system. Who has one, who doesn't have one. There were nerve nets, and then there were nerve ladders, and then there were ventral nerve cords until you got to the vertebrate nervous system, which has a dorsal tubular, meaning like hollow nerve cord filled with cerebral spinal fluid, right? Centralized. And then we went through and we looked at the different regions of the brain. And so the hind brain, when you look at that, those are really important because that's what keeps you going without thinking. Would that be part of your autonomic nervous system or your somatic? Autonomic. Would it be parasympathetic or sympathetic?
Sympathetic.
Both. Okay? Yeah, it was. Sympathetic if you're trying to what? Under stress, right? Parasympathetic, normal, right? Which one would digestion be? Sympathetic or parasympathetic? Parasympathetic, right? If you have time to digest, you're not under stress. Which one would be playing a soccer match? Sympathetic, right? Now, would you also have to enlist your somatic nervous system? Yes, to move your skeletal muscles, right? So you might want to review that whole overview. There were several overview slides I gave you and told you to look at, and I can hit those as well. Okay, but anyway, back to unconscious activity of the hindbrain. The key one here, medulla oblongata, heart rate, breathing rate, vomiting, all of those types of things. Also part of the hindbrain is the cerebellum. What is the cerebellum? Is it your terms?
No, to make up the quiz.
Oh, you know what? I didn't set that up. Which quiz is it that you need to do?
The endocrine system.
Okay, you know what? Hang on one second. I don't know if it was when we did the quiz. So no, you don't have to worry about making it. Because I have to throw it out. If I end up using it, I'll let you know. Okay, come back to me. Sorry. What is the cerebellum about? Balance. Also,
Muscle tone.
yeah, muscle tone and coordination as well. Okay. To fine tune what your frontal, your cerebrum told you to do. Okay. Okay. And then midbrain is kind of relay station. That's easy to remember because it's right in the middle. And then your forebrain, this is thought, memory, learning, cognition, metacognition, thinking about thinking. Thalamus and hypothalamus, right? Now, hypothalamus, do you have to think about what the hypothalamus does? No, so that would be more ANS, right? Hypothalamus is true nervous tissue, yet it controls the pituitary gland. Okay. This is a good diagram. There's several in there. Breaking down, here's your central nervous system, here's your peripheral nervous system. Central nervous system is just your brain and your spinal cord. If you had to come up with one word for the function of the spinal cord, what would you say?
Reflex.
Exactly what I would say. Boom, shakalaka. I would say reflex.
What did you say?
He said reflex.
I wanted to say it quiet enough so she would hear it if it was right. And
So you wouldn't be mocked?
Yeah.
Okay. Reflex is what I would do for spinal cord. And then brain, controls, moderates your entire environment. On your peripheral nervous system, how many spinal nerves do you have?
Pairs. 31 pairs.
How many pairs?
31 pairs.
Your spinal nerve, you have 31 pairs, and they are all what? Oh my God. Okay, they are all mixed nerves, which means they have both sensory and motor. Okay? How many cranial? 12 pairs. Some are sensory, some are motor, some are mixed. Okay, got that? Okay. Sensory nerve fibers are going towards the central nervous system, where motor is going what? Away. Of those motors, some are going to somatic. If I was going to come up with one word for somatic, what would it be?
Effective.
Like movements.
This is the word I would come up with.
Choice.
Choice. Right? You choose to move it. These are, the target here is your skeletal muscle. That's you choosing to move. Is it ever that you don't choose, meaning like you're not consciously thinking about it? Sure. When you're doing a what? Reflex. Okay? Autonomic, what's the word I would think of for
Organs.
autonomic? Automatic. Yeah, I would think automatic.
Organs. Organs are autonomic.
Okay? And then you have two divisions of this. Sympathetic and parasympathetic. Give me one word for sympathetic.
Stress.
Exactly. Give me one word for parasympathetic. Relax. Okay, so this information is going out via the motor nerves. Okay, out. And then sensory is coming in. Okay, any questions on that? Yes.
Those two numbers are important, correct?
I just put them up there because I couldn't think of anything
That was a dumb
else
question,
to do.
I'm sorry.
Okay, all right. Then we looked at our mammalian brain compared to our lizard brain, and I told you to stay in your mammalian brain. Your lizard brain is paranoia, food, sex, pleasure. There's some learning in there. Okay, let's see. Maybe we can do this as a review. You can tell me who matches what, okay? So I'm going to pull one. I'm going to pull CNS. Brain and spinal cord. Good. Okay, we won. What's Hydra? Can you... Here, I'll pull it so you can see this on your computers. Does it say hi? Okay. Genghis Khan. Okay. Okay, now you can see it on your computer. What would you do for Hydra? I agree. Oh, dragon. Sponges. No nervous system? Do you see it?
Yeah, no.
Oh, no nervous system but responds. Good. How about has folds and crevices?
Gray matter. Higher mental function.
Higher mental function?
Yes, I would say that.
Yeah, because by it's creating more what? Surface area. Good. PNS.
Peripheral nervous system.
Okay, so there's not peripheral
Outside of
nerve
CNS.
outside of CNS. Good. Okay, let's do Hind Brain.
Most ancient.
Is this a most ancient? Okay. Planaria.
First.
Start of cephalization. Okay. Midbrain. Earthworm.
First CNS.
Okay, and forebrain. Process sensory info. Good. All right, muy bueno. Now, do you need to know the parts of the neuron? Yes. What part sends? Axons. Axon away. Axon away. Okay? Axon, axon. So axon is away. What does the myelin sheath do?
Code signal.
Insulates. Good. Okay, what do the dendrites do? Receive. Good. Axon terminal button. It's part of the synapse. Give me more. Yeah, it could connect to a dendrite. What does it contain? Neurotransmitters. I didn't hear you. I'm sorry.
Neurotransmitters.
Thank you.
Got it.
All right, good. What does the nucleus do? What could it do based in this situation?
Signal.
Process? Sure. Okay, and the soma just contains the nucleus. Yes.
So within the neuron, the signal's electrical, but between the neurons, it's chemical, correct?
Exact. Okay. What else? All right. So then we talked about not only are there neurons, but there are support cells that help take care of the neurons. They could be phagocytic, getting rid of debris. They could be like astrocytes and bringing in nutrition. It could be about repair. Schwann cells out in the peripheral nervous system, oligodendrocytes, right, within the central nervous system. They could be creating cerebral spinal fluid. Okay, so the idea there is that there are the neurons, the conducting cells, but they have a posse of support cells as well around them. Okay, and here we already went over all of this, so you should know all of these. I believe we just reviewed it as well. Is there anything more? Okay, Baba. All right, any questions on that part? Oh, pictures are happening. Okay, along the length of this neuron in yellow, what kind of stimulus is that? You already told it to me.
Oh, the electric.
Electrical. Right here at the synapse, it'll be what?
Chemical.
Chemical. Good. All right.
Yeah. Got it.
Now, what makes the electrical travel faster is when it can leap from node to node to node of Ranvier, and that's what the myelin sheath does. Insulates and speeds up conduction.
So the myelin sheath is like multiple little chunks of it. It's not like one long connection, right?
It's one cell kind of wrapped around it.
It's like a single cell, like multiple
Yes,
cells.
one Schwann cell for each area here, kind of wrapped around it. Okay, lava. This was just showing you some of the different cells involved in the central nervous system. Then we went here to types of neurons. And what I want you to see is if the cell body is somewhere where you're receiving information, then there's some sort of processing that's going on there. That's where you're going to get, and here's a good word to know, integration. Integrating all the yes votes and integrating all the no votes. And then coming up with a summative, because a neuron doesn't kind of fire. Okay, it's not like kind of motivated, like, okay, I'll go. It goes the same speed. Okay, it either fires or it doesn't fire. It's an all or nothing. Okay, and it fires at the same speed every single time. Okay, a neuron can't move any faster than its standard speed. You know, whatever it is for that. You know, if it has a, you know, myelin sheath, it all obviously goes faster than if it's, you know, unmyelinated. But the speed is the same. It either fires or it doesn't fire, all or nothing. In order to trigger it, to make it fire, you have to reach the threshold of depolarization, right? Okay, you have to make it less negative, a little bit positive. Usually what's the threshold? Yeah, negative 40 in there somewhere. You're going to have, you hit that threshold and where the sodium gates start flying open. You look at this type of neuron right here. So you have dendrite, it's going through here to this axon. In this case, most sensory neurons look like this. This cell body is just kind of, its job is to maintain the neuron, but it's not processing any information. It's either been triggered or it's not been triggered. Okay, and we looked at this. Okay, so sensory neurons go towards, motor neurons go away. In your autonomic nervous system, you have two motor neurons. Two motor neurons in your autonomic nervous system. Okay, and remember your autonomic nervous system is going to like your, it could be your capillaries where you have smooth muscle, right? Those sphincters around the capillaries. It could be going to your stomach, to any kind of gland, and to your heart, right? I said, can.
I was just wondering, how come the myelin sheath makes the neuron?
Because it only has to depolarize and repolarize that one little section instead of having to maintain the whole neuron. You know, and having the whole neuron charge, you just have to focus on the exposed areas. It's kind of like if you had company coming over but you knew they were going to stay downstairs, then you only have to clean the downstairs. I'm just saying.
Not according to mom.
Not according to mom. The whole house has to be clean.
Clean your fucking.
That would be me
It's
too.
like examining your closet.
And then you say stuff like, nobody's going to see it. And then your mom says, but I know. Is that right? We get that in the mom handbook.
She's not that nice, but.
She's not. Oh. All right. So this is a typical motor neuron.
I have a Korean.
Typical sensory neuron, typical interneuron. And then we saw here, I gave you the example, like you step on a tack, you have the dendrites receiving sensory information, synapsing here with an interneuron. This could be in your brain or your spinal cord. Synapsing here with a motor neuron to pull away. All right. Skip the monster quiz we did in class. So now let's talk about this. If I said resting potential, you would tell me that's when a neuron is what?
-70.
Okay, -70. What else could you tell me?
Positive on the outside. Negative on the inside.
Okay, it's not firing. That's what I was after. Okay, resting and not firing. Okay, now tell me what is the resting potential? It's around -70 millivolts. There are 3 things that contribute to the resting potential. Don't you see that as like a perfect essay? Just if I said, tell me 3, parts of the inflammatory response. Okay, you wouldn't tell me sodium goes out and then sodium goes out, and then the third one is sodium goes out. Okay, so what are the 3 different items that set up the resting potential? Sodium, potassium, pump.
The ions.
Okay, wait, so let's deal with that one. Sodium, potassium pump, which pumps out 3 sodiums for every 2 potassium. Now let me tell you, if this were an essay question, you would tell me the sodium, potassium pump. A pump is a protein carrier that requires ATP. The sodium potassium pump pumps 3 sodium ions outside of the neuron and 2 potassium ions inside. You wouldn't say to me, if I said what sets up the rest of it, sodium potassium pump, okay, you would tell me what that is. You would explain and tell me its significance, what role it plays. Okay, so that's good. What else besides the sodium potassium pump? You are all saying them.
Negatively charged ions.
Yep, negatively charged ions and proteins on the inside. What else? Leaky potassium. Whoa, you leaky potassium. So potassium leaks back out. So the outside has more positive charges than the inside has positive charges. So there's a differential right there. On top of it, you got negatively charged things inside. And on top of that, potassium tends to leak back out. Now, the sodium potassium pump is constantly working. The ions are always going to be in there, so you don't have to worry about that. They'll always be in there, and the potassium gates are always going to be leaky. That sets up your resting potential. Okay? If I am trying to get a message from my foot to my brain, how am I going to do that quickly? I'm not going to do diffusion of a chemical for it to diffuse all the way up my foot and to my brain. I'm not going to use my circulatory system because it's going to have to return from my foot in a vein to my heart, to my lungs, to my heart, and then to my brain. And it's not like all of it would be there. It would be very diluted, right? So I want to do something quickly. So how can I quickly communicate from one part of my body to another part of my body? Because I'm a large multicellular organism. And I need to respond. And I not only need to respond quickly, but I have bilateral symmetry. So if I'm going to lean to protect myself here, I've got to counterbalance over here, right? So all of those things have to be coordinated quickly. So how do I get information from point A to point B? Okay, so somehow I have to respond by some sort of chemical. Now, could I do it just electrical? Could I undo a paper clip and jam it in this socket right here? Yeah. Would that work? Yeah, because my neurons are all nerves. So they will all fire, and at all the synapses, they will release acetylcholine out at my skeletal muscles. And when I jam that paper clip in here, my muscles will all contract, and I won't be able to remove my hand. Right? That's what happens when people get electrocuted, unless it's like really high and it blows you away. Right? You can do that. But otherwise, you can't get away from it, and that's why you need somebody to like run and tackle you. You don't want to go, no, because then you'll be, right? And so you don't want to do that. So you would have to like hit them with a stick or run full bore and like run into them and break them. Okay? Little tip in case that happens to you. Okay?
He's writing that down.
No, pour water on them because that helps cool off the electricity. Okay? No. Okay. So what happens then is whatever chemical lands on here starts to open up some sodium gates. That does not mean this neuron will fire. Enough have to open until it reaches the threshold. Then the light is green and all the sodium comes in all the way along. Brrr. That is the action potential. That's how a neuron fires. That's it. Sodium gates from one end to the other open up. Okay? And then you already know what's going to happen here. At the end, what kind of gates are going to open up? Calcium. And then calcium comes in here at the end, and this causes the synaptic vesicles to fuse with the presynaptic membrane and release its neurotransmitter across the cleft, which then can bind to the next neuron or muscle or gland, right, and trigger a response in that, if it's excitatory. If it's an EPSP, excitatory postsynaptic potential, right? Okay, so now if this neuron wants to fire again, how am I going to reset the neuron so it's positive on the outside and negative on the inside? What's the quick fix?
Potassium on the outside.
Yeah, potassium goes out, but ultimately, if this neuron keeps firing again and again, more and more sodium comes in, more and more potassium goes out, I'm not going to be able to work. Those voltage-gated channels are specific. It opens so sodium can move from here to here, not the other way around. So the way I need to fix that is that my long-term fix is the sodium-potassium pump, which pumps 3 sodiums back out and 2 potassiums back in for my long-term fix of the problem. Yes?
So when you have potassium on the outside and sodium on the inside with a short-term fix, it can fire again still?
It can fire again. What I'm not showing you in this picture is that there's a lot of sodium out here. And there's more potassium in here than what I'm showing you. So what happens is the ones closest right here come in. That's it. The gates are open for a very short time. And potassium goes out there and it just kind of realigns so it's positive and negative on the inside so it can get depolarized again to trigger an action potential. Otherwise it wouldn't depolarize,
so you need
right?
the positive on the outside and the negative on the
Yeah,
inside.
that's your big thing. Yeah. Yes?
So when does the membrane potential hit like positive 40 volts?
When enough positive comes in to counteract the negative, and when it raises up, then it will trigger it.
Okay.
Yeah. All right. So if you look here, okay. So this is showing you a resting potential of minus 65, minus 70. That's fine. The outside is positive. The inside is what? Negative. Okay. You can see inside the sodium is orange and the potassium is pink. Inside here, is there more positive or more negative charges? Oh, that wasn't a good question to ask you. Is there more sodium or more potassium? More potassium on the inside, okay? And if you look on the outside, okay, so orange is sodium. There's more sodium outside, right? So that's the resting potential. So then next what happens is you start to get some depolarization and the gates open. So you have this quick reversal of charge right here. And so sodium starts to come in and then potassium will go out immediately after. So here we're up to plus 40 right there, okay, in order to reset it, but you will use your sodium potassium pump all along. Here is a little nothing. Took 10 years.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed and the inactivation gates are open. Voltage-gated potassium ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
Okay, so there is that refractory period which keeps the impulse going in the same direction. Any questions on action potentials and how that works? Yes?
So the long term kind of includes the short term, but it's like that extra step with the sodium potassium pump, right?
No. The short term is the potassium rushes out. The long-term fix that you need to maintain neuron activity is the sodium potassium pump. All right, so take a look right here. This is a really good graph. Understanding this graph, that's the kind of question they would like to ask you. You know, explain why it has this resting potential. Why is it changing to positive? What has occurred? If they say, if you have something like this, they could say, what occurred to set up event where it's at A? What is occurring between A and B? What is occurring between B and C? You see what I'm saying? And you know, we already talked about what sets up the resting potential. Why does this become depolarized? What's happening?
Sodium rushes.
Sodium's going what? In. Good. And over here? Potassium's going out. And so much goes out, that's that refractory period to keep it going in the same direction. All right, then that brings us to the synapse. Electrical along the length, chemical, where two neurons meet, right? And so you already know this, you told it to me. Calcium gates open. In fact, we could use this. Calcium gates open, right? Sodium was going in, now calcium goes into the presynaptic neuron. Calcium causes the synaptic vesicles to fuse with the presynaptic membrane and release its neurotransmitter. Something like acetylcholine we talked about, it doesn't act the same everywhere in our body. It's excitatory out in our skeletal muscles, but inhibitory at our heart. Okay, but there are neurotransmitters. Some in the brain, dopamine is another one. It's a neurotransmitter. Okay, and when that neurotransmitter is released from that synaptic vesicle, it's just pure diffusion, going from the presynaptic membrane to the post. There at the postsynaptic membrane, it will bind to a receptor on the dendrite, and they're usually ligand-gated channels, not voltage-gated channels that change due to voltage. They change because something landed on it, okay, and opened it. And then if you want it to be an excitatory postsynaptic potential, sodium comes in and can depolarize potentially that neuron to trigger another action potential. Okay, if you don't, then it's going to hyperpolarize it. You know, it's going to do something to keep it more negative on the inside and more positive on the outside. And then what a neuron has to do is it has to add up all the excitatory and inhibitory messages, which could be 10,000 messages that it has to add up. And those closest to the axonal hillock are going to have, where it starts to become the axon, are going to have the greatest impact right here, closest to where you would trigger your action potential. And those neurons, what integration means is adding up all the yes and no votes. And then you go with whatever it is. Okay, but it either fires or doesn't fire. Same amount, same speed every time. Just won't fire if it doesn't reach the threshold. Okay, questions on that you want to ask me? Okay, then we move into the central nervous system. Central nervous system is brain and spinal cord. It is protected. What is it protected by?
The fluid, the
Cerebrospinal fluid, that's one thing. Give me another protective. The meninges, the pia, the arachnoid, and the dura are all protected. What else? The bones, exactly.
Yeah.
Yeah, your skeleton, right? Your spinal column, you know, is there protecting your spinal cord. So the bones, the meninges, and the fluid all work to protect. Right? And we took a look at the different membranes, looked at the cerebral spinal fluid. Okay? Then we're looking at the spinal cord, and in the brain too, you have gray matter and you have white matter. Okay? It's all important, but what is probably more important? Why? I agree with you. That's where you can make what? Synapses. The more synapses you can make, more pathways you have laid down, the more likely you are to make connections, right? And to understand something. Okay? So, and I'm going to tell you, I don't know if I remember talking about this at the beginning of the year, I might have, and I haven't finished the book, which is annoying. I really need to finish it. But it was a book talking about how all of you could be excellent mathematicians and engineers. And I don't know if you ever experienced, I certainly experienced this in school or in life in general. You try to solve a problem and you can't think of a way out, but then later, when you're not thinking about it, you're like, ah, and you have those aha moments. Have I talked to you about that? And then after you take the test and you send me a message, I didn't mean that. Okay. So I'm going to tell you, it's a skill I want you to start doing. What happens is you think there's this one way to solve it, and you sit there and you can't figure it out like a math problem, and you just keep going around and around that same way, bumping into the same wall. And when you relax and stop trying to make your brain use that neural pathway, your brain is amazing at solving those problems. Okay, do you need me?
I was actually looking for Miss Chevalier, but I thought maybe she'd be in here.
She is not here.
Are you sure? Did you check under the tables?
I don't see her. No problem.
Sorry.
When you relax and I know it's hard in a test to mentally do that. Leave it alone, and that's why I say if you have a hard problem, what should you do? Skip it and come back to it, because you will be amazed at your aha-ness, and you can actually practice being better at that. Because some of you, what I get so frustrated for you, which I know you're more frustrated than I am, when you study something and you know something, but you just can't pull it for the exam. Right? Or the quiz.
Me and class.
Right? So, back away from the way you think you need to do it. Let your brain relax, work on something else, and come back around, and you'll start to encourage. When I know I've reached a hurdle and I think, how am I going to do this? I have to be gone on this day, and they have this lab on this day, and how am I going to manage this? If I sit there and just try to force issue, but if I relax, I just go, I know my brain's going to come up with something awesome. I'm just going to leave it alone. It's going to come up with something. You'd be surprised how many times it will come up with a resolution for you. You know about neurons now. Stop trying to force yourself to go down that one neural pathway. Allow your brain to try some other circuits, which might get you to your end zone. I'm just saying, just try it. It can be personal, it can be in life, it doesn't have to be necessarily school. Okay? Because now you understand what thinking is, right? Okay? So here's this gray matter, here's the white matter. And then when we look at a spinal cord, we can see it here, we can see the membranes, we can see the bone. But what I really want to go here. Okay? Sensory information always goes in your back of your spinal cord. Sensory information. What are you doing? Did you say something bad that I don't like? What'd you say, Anish? What? Okay. All right. So making Teju choke. Sensory information comes in the back. Okay? And then what's going on? Okay. We'll just leave it alone. Synapse is with an inner neuron, and then that will synapse with a motor neuron who will exit out the front. Now, in reflexes, at the same time that it synapses with a inner neuron, it's also synapsing simultaneously with a motor neuron. It doesn't count when you say it after I say it. Okay, and then when we look at our brain, okay, but I appreciate the participation. Right here, we know what this does. What does this do? Thinking, memory, learning, thought, right? Thalamus, this is when you have memories with emotions. Good. Okay. Here you have your hypothalamus, which connects your nervous system with your endocrine system. Here's your pituitary. Midbrain, we're fine. Pons, we're fine. This is key. Medulla oblongata. We know what that does, right? Okay. Cerebellum, right? Okay, so just review those. And then peripheral nervous system. Here you can see the different pairs of nerves. How many nerves are coming off the brain? How many pairs? 12 pairs. Good. Okay, and then 31 pairs coming off the spine. Remember, sensory going in the back, interneuron, motor neuron out the front. And then I want you to think this diagram right here. You good on this one?
Sure.
You all have this? Okay, and then this is a really good one too, right here. Okay, that's another good one to have because it summarizes it. And then like I told you here on your autonomic nervous system, you have 2 motor neurons out to your target. The sympathetic nerves come out in the middle here, and they synapse. Here's neuron 1, and then here's neuron 2. Neuron 1 synapses right away with neuron 2. Right outside, it's called the dorsal ganglia. Whereas in the parasympathetic, the first motor neuron is very long, and then the second neuron is short. So where they synapse is different, and that's probably a really good strategy. So you don't, have mixed signals because the synapses are in two different locations. Does that make sense to you, evolutionarily speaking? You could support that, like as an adaptation, that they synapse in different places, so you don't cross-mix your excitatory and inhibitory messages. And that's it. Good job, you guys.
Cool.
Super smart, super smart.
Do time for the
Quiz
test.
tomorrow.
So,
Time for what
to
test?
see.
Oh yeah, sure, you can look at them, that's fine. And have a piece of toast if it's late, but don't stay up too late because your brain won't work tomorrow on your quiz.